Спасибо вам
Thank you
всем.
all.
Я понимаю,
As
что
I
критическая масса
see,
уже у
the
нас
majority managed to
созрела
come
для того,
back, so
чтобы
we can
продолжить нашу
continue our
работу.
work.
Спасибо еще раз.
We
Мы
start our second
приступаем к
session.
обсуждению вопросов, включенных во вторую
I want to
сессию
inform you on behalf of
сегодняшнего
organizers
дня.
about some
И я
changes
хотел
in our
бы
agenda.
от имени организаторов проинформировать вас о
First
частичном
of all, taking into account the
переформатировании
continuity,
нашей сессии. Мы имеем в виду, что мы с учетом преемственности обсуждений, которое так или иначе было начато этого вопроса вчера, пригласили Алекса Кудрина для того, чтобы он поучаствовал в качестве сомодератора также. Елена Анатольевна, я думаю, возражать не будете. Тем более, что Алекс имеет неоспоримое преимущество в силу того, что нам надо определиться с понятийным аппаратом, как мы вчера говорили, потому что очень много наносного, надуманного, я бы сказал, или даже бездумного, как сегодня выразился один из докладчиков, кстати, российских относительно вопроса, который предстоит обсуждать. Поэтому, Алекс, персональная просьба к вам прокомментировать с точки зрения подходов Европейского
Union in the issues of substitution, interchangeability, etc. Irina Anatolievna, it would be nice if you give the floor to Alex so that we start with the definitions from the very beginning. It would be really great.
Поделились понятиями, то было бы замечательно с самого начала. Спасибо.
Спасибо, Владимир Григорьевич. Конечно, я думаю, что все-таки, Владимир Григорьевич, можно мы, может быть, пойдем по повестке. Я обязательно Алексу предоставлю слово. Не хочется оказывать пока давление на докладчиков с тем, что они будут произносить, да? И как бы я думаю, что в любом случае мы должны резюмировать то, что будет здесь происходить, у кого какие подходы существуют в различных странах. Моя задача сегодня самая выгодная. Я буду в основном только следить за таймингом, а все остальное, касающееся уже и содержательной стороны, я надеюсь, что у нас останется время, как по регламенту положено, и на дискуссию.
Time for discussion, for clarification of unclear issues, because following the things that I know from psychology, if we need people to memorize something important to keep in mind, in memories, it should be actually spoken in the end of our session. So finally, I think we can summarize, and everybody will be leaving this hall with the formulation that will be delivered in the end of the session. We're discussing very complex issues. Yesterday I tried to focus on two aspects. One of them is the economic part of interchangeability. The second issue is the therapeutic aspect of interchangeability, because in reality, in Russian Federation, I want our foreign colleagues to know also it's one of the key issues, because we should make decisions on interchangeability only during the procurement of drugs following INN, but also following the issue that here in Russia our physicians have to prescribe drugs following INN. There is always associated with the issues of adequate therapy, adequate pharmacovigilance, adequate interchangeability from therapeutic point of view. And I also have a question
У меня будет еще
that I
один
want to
вопрос,
formulate
может
in the
быть, кто-то
beginning
из регуляторов,
of this
вот его я
session,
задам в
because it's
начале.
interesting for us to
Нас
know
очень
how foreign
интересует,
regulators
как
manage
все-таки
to
нашим
limit the
зарубежным
number of positions,
регуляторам
number of drugs
удается у себя
which have
ограничивать
the same
количество
INN, because
позиций
here in
в
Russia
рамках одного
we have
международного
to deal
напатентованного
with the fact
наименования.
that
Потому что в
for
Российской
each
Федерации мы в
INN
настоящее
we
время
have
сталкиваемся
more
с тем,
than 100
что на одно
generics
непатентованное
reproduced
наименование мы
drugs.
имеем более сотни
It's a huge number, as
воспроизведенных
you
лекарственных
understand, and
препаратов,
you
дженериков.
really
И
can
это, конечно,
imagine
огромное количество, и я думаю,
all
вы
issues
понимаете, какие
associated with
сложности
this.
возникают в связи с этим.
This is my watch. I put it here, and I hope to stick to my time. I will do my best. So, first of all, good afternoon to everybody, ladies and gentlemen, and thank you, Madam Chairman. I would like to thank IFPMA, AIPM for inviting me, the government or the authorities of Russia, and in particular, because behind acronyms there are people, I would like to thank Ms. Janice Bernat and Ms. Chiara Gherardi, because they really did all their best to having us here this afternoon and the entire conference. I am the INN program manager. I have been in this position since 2001, quite a number of years, but I'm not yet bored. And the more I do this work, the more I find it fascinating. I feel like I am the outsider in this group of experts because I am not at all an expert in biosimilars. But having worked with you and with other colleagues for a number of years, I had the chance to learn, and I still had the chance to learn these 2 days. And I find this adventure of working in this area even more interesting day by day. Okay, I can see it here. There is the introduction to my presentations, which is not mine, but is the one that our Director General wrote. I will not go through, or perhaps I have to because of the Russian translation. It is written that some activities undertaken by WHO are largely invisible, quietly protecting the health of every person on this planet every day. By assigning a single international name to drugs, WHO helps ensure that a prescription filled abroad is what doctor ordered back home. So, the mandate of clear identification of what is there, and it has been recognized very strongly, even by our Director General, and very recently. Just some basic facts of WHO, because I don't know if all of you are familiar with what WHO is. We have 193 member states, 2 governing bodies, the World Health Assembly and the Executive Board and the Secretariat. One of the articles of our constitution says that WHO is responsible for international standards, and the name is certainly one of these. The program of the INN is, I think, the oldest program in WHO. It has been initiated in 1950. WHO exists since 1948, so it's really very, very old. And it became operational in 1953. It is based on WHO constitution, and we named the first biological long time ago. What you see in these parentheses numbers are the list number, and the first one is the proposed number of the proposed list number is 48. We are now naming 110. So it's quite a number of years ago, if you consider that we have 2 lists per year, that we have named our first biological. WHO has indeed the responsibility to develop, establish, and promote international standards for pharmaceutical and biological products. And there is a need for a universally available and accepted name for drug substance. If the name is not accepted, ça va sans dire, it will not be used by anybody. INN is a unique generic name that recognized that it is recognized globally, and it is formally placed by WHO in the public domain. It doesn't belong to anybody, including the applicant for this INN name, but it belongs to the entire world. And I believe that if there are pharmacists or medical doctors among this audience, I'm sure there are, the majority of you studied using INN and not using trade names. It is intended for use in drug regulation, prescriptions, pharmacopoeias, labeling, advertising, scientific literature, etc., etc., etc. We don't work alone. We have many interested parties outside WHO with whom we work, regulators, national trademark departments, the research-based industry, other nomenclature bodies such the USEN program, the pharmacopoeias, groups that were initially not in the scope of the program, like the World Custom Organization, but now we work very closely with them because funnily enough, or interestingly enough, they base their own classification of medicines and their own fee classification of medicines on INN. So they need our advice and we regularly help them with this. What there is in a name. I have seen that Dr. Maxine Kina mentioned the discussion regarding some definitions before, because indeed a name is just a name, but there is a lot in a name. And in particular regarding the medical field or the pharmaceutical field, what is safety, efficacy, rational use, regulation, procurement, information is linked to this name. So it's not just a name, it's something more than a name. What is an INN? It's a unique name. We said it. It should be distinctive in sound and spelling from other INNs or trade names or all sorts of names in common use. It should be not liable to confusion with other name. It is formally, I have already said, by WHO in the public domain, and it can be used without restriction to identify pharmaceutical substances. The system, how do we function? It's a very simple system. We are a very modest WHO secretariat based in WHO, and we could do nothing without our group of experts. And among these experts, I am sure that the majority of the people in this group knows very well Professor Galina Shashkova, who is sitting in front, and she is a kind of cornerstone of the INN program, representing all the Russian-speaking countries, not only Russia, but the other countries that speak Russians, and she has been in the program before me. So I think that she knows much more than me, and I'm very glad to see her sitting here. So if I say something which is a mistake, I'm sure she can amend, correct, ameliorate what I said. If it is needed, we can call on further experts, and some years back, when we realized that the problem of biologicals was becoming a big one, not only for biosimilars, but for all kind of biologicals, we decided that we need to formally establish an advisory group on biologicals to help our experts, who are experts in different fields, from nomenclature to regulatory background, pharmacologists, biochemists, chemists, pharmaceutical chemistry expert, etc., etc. But they are not only only focusing on biologicals, so we wanted to give them some support, and we have established this advisory group on biologicals. And of course, our part of our system is the publication, because what we produce we have to disseminate through formal publication. When we publish a name, all the member states receive a circular letter through us from the Director General, and the Director General formally informed the member states that this is a proposal. Should no objection be raised to this proposal, then this becomes a recommendation. And of course, times evolved. Not only we stick to this formal system, but we also publish everything through different electronic system, not only web, but we have extranet, we have all sorts of facilities to have access to the INN data. This, I think, it's an old slide, but I like it very much because in 2005, I think that it was shown that although there are only 20%, maybe this is also, the data are changed, of the medicines that on the market are biologicals, if you consider the new medicines in development, the situation dramatically changed. And when we well, this is dated 2005, so perhaps a few years back, we realized that this was in clear, that was an evidence. Then we decided that we need to put some of our energies into the naming of these biological medicines. And this is just to show you that in 2006, perhaps 5 to 10% of the INN requests that we were receiving were biologicals, and now the trend is expanding, and 40 to 50% of the requests that we are naming are biological products, a huge number compared to the past. What is the current position of the INN group regarding the biological products? We have 20 biological groups with STEM, some with 5 groups with a more formal nomenclature scheme, sometimes very complicated, like the one for monoclonal antibodies. We have developed general policies, and we have created the book, which is kind of repertory, which is an INN for biological and biotechnological substance review. This is the address where you can find all the information of all the decisions that have been taken. It is a living document because each meeting we change. We change our policy, we name more products. We are currently debating the possibility to name cell therapy. So it's not something that we stick to this and it is like that. It's evolving with science, with knowledge, with you. And okay, this is just to show that some products are excluded from the system. The blood products are excluded, the immunoglobulin, skin substitute, most of the vaccines, but we name what the applicant called therapeutic vaccines. We have named some of that, but not the classical vaccines, of course, because these go to the ECBS. And we are discussing, and I think that some conclusions have been taken by the expert group, to name cell therapy products. This I will not comment, but it's just a visual slides, just to say that many of these stems, so the parts that identify they belong into a certain groups, you have been using for ages, MAB, COG, Hermine, Parin, perhaps without even knowing that these were coined and invented by the INN expert group, but it is such. There is an issue which maybe is of interest to the audience, is what do we do for glycosylated proteins, peptides? This is what we have done until now. We have identified the group with the stem, poietin. We have indicated a difference in the amino acid sequence, if any, through a prefix. We have indicated difference in the glycosylation pattern with a Greek letter spelled in full as a second letter. Everybody is aware of apoietin alpha, beta, gamma. Of course, we have not been very consistent. I will not go into this with the use of the letters, I mean, but this is what we have done. And we have started to do this 30 years ago without having the knowledge that we have now and publishing just glycoform alpha, glycoform beta, because the experts recognized in the past that there was a possibility for a concern and they wanted to be somehow conservative to protect patient safety. And the Greek letters are assigned in alphabetical sequence as applications are received. The concept of biosimilars, we had a meeting in 2004, if I remember correctly, or 5. And we have published these considerations that are our current position. The concept of biosimilar, very important for us. It's a regulatory one, not a nomenclature one. Generic substitution and reimbursement are health policy matters, not decision taken at the WHO level. Decision on interchangeability is not done by WHO. INN are part of the pharmacovigilance system, not the only part, but a very important part. There is no specific INN policy for biosimilar, and I have written currently, because there is a lot of discussion in the group currently. And then glycosylated protein from different sources expected to different in their glycosylation profile are given a different name. The same approach is used for all post-translational modifications. Please remember that nevertheless, WHO system for having an INN is a voluntary system, and WHO in no way can force applicant to apply. This can only be done through a very strong and important collaboration with national regulatory authorities. However, we realized that there was an issue with naming of biosimilar or similar biotherapeutic products. And we started from that, but I agree with Thomas that we should expand from that, because it's not only SBPs. There has been discussion in the group saying that WHO program exists to facilitate the identification of medicines so that they have a unique name globally recognized and that the medicines is rationally prescribed and used. At the present, SBP are not specifically named, but general naming principles are applied. In practice, this means that some SBPs, all unglycosylated, so simple amino acid sequence such as filgrastim, somatotropin, have the same non-proprietary name as the reference substance. Glycosylated SBP are likely to have different non-proprietary name to the reference. The different interpretation of this policy has led different countries having different non-proprietary name for the product. Unfortunately, we have one case that I think is known to everybody of an epoetin that has been approved as epoetin alpha in Europe, but has not been recognized as an epoetin alpha in Australia, and as a result, an INN name is used for this epoetin in Europe, but in Australia it is called lambda, and this is not an INN, it's a TGA name. It's an unfortunate situation that we want to try to prevent in future. When different non-proprietary names are used, the reference substance to which the SBP was compared is not identified. And this is another issue that we have. We have cases where the reference substance is one, but they have applied and they've got another name, so they are referring to a reference substance, but they are having two different INNs. This is just a visual one, which I will not comment very much, but you see, if we have generics chemicals, the same INN as the reference, and this is simple. We have then, I would say, at least two type of SBP and one non-SBP. The first type is highly similar and cannot be distinguished from the reference by analytical quality testing. The amino acid sequence is the same. The post-translational modifications are highly similar and the substance is comparable, and this substance is likely to receive the same INN as the reference. But we have another type, the SBP2, type 2, which is comparable to the reference, but has significant differences in analyzed parameters. And, okay, the amino acid sequence, of course, is the same. The post-translational modifications are similar to different, not highly similar. And in that case, the same INN stem and name with a Greek letter is likely to be assigned. And then we have the non-SBP, which is, well, a bit different or more different, and most likely will receive a different INN. Oh, the situation is very confused because now everybody is doing whatever they want, and we realize that regulators, to defend themselves, are doing many different things in many different national settings. So when we discuss among the group the need for naming SBPs, they raise this issue. It is the near universal opinion of regulatory bodies that switching arbitrarily between SBP and the reference product, or SBP and SBP without, or between SBP and SBP without a medical supervision is undesirable. I think that everybody agrees to this. Does it mean that we have to distinguish an SBP from another? As prescription by non-proprietary name is common, and some countries encourage this, and WHO does as well, the issue of naming SBPs does affect the INN program. And this is the opinion of the group, not only of the group, of the majority of the group, because it's still under discussion this, but of some regulators that the group has referred to for advice. The major regulators, obviously. So the naming of an SBP is an INN issue. We think that in two ways the INN program is affected. In first instance, if the reference and SBP INNs are the same, it may make substitution of SBP for the reference substance more likely, certainly not less, and worse still, substitution for one SBP by another. So there is a risk, as Thomas highlighted. The risk is there in all its splendor. If individual regulatory bodies develop, and it has already happened in many countries, Japan, Australia is likely to do, Singapore, I don't want to comment on what they are doing in USA, but there is a lot of happening in this field. But if individual regulatory bodies develop their own naming policy for SBPs, there will be a diversity in name and not a unique name which is globally recognized. This goes against the mandate of WHO. So there is a proposal, still is a proposal, still is under discussion in the group. It is therefore, well, what they are discussing, the expert, is that all SBPs, and I think we are starting from there, but we will go beyond this, should have the INN as a reference product, as a first part of the name. This is effectively and unambiguously identify the reference product for the SBP and identify what is what. But there should be a second part of a name, but that perhaps will not even be a formal part of the INN, because we are discussing how to do, that should identify the medicine as this one is this one, uniquely. We haven't the final solution because there is a lot of discussion, and perhaps this will be addressed globally through a database, a publishing of an amendment, an annex, not an amendment to the list each time, and certainly should be done together with the regulators. But we are at this level now. We need to know what is what. We need, for the reasons that I mentioned. There has been some discussion, as I said, on how this should be implemented. And okay, I try to summarize because the discussion was very broad, and now we are to these two options. The second part of the name should be assigned by the INN program according to an agreed policy, and I believe personally that this should be the mandate of WHO, and we should be responsible for that. Another proposal that was not favored, but it is a proposal, is that the INN program produce an advice document laying out an agreed name scheme which may be applied by all regulatory authorities. I'm going beyond my time, sorry, but I'm concluding here. It's really just to give the flavor of the discussion, and the conclusion is that the naming of SBP is an issue that does need to be addressed globally. And I agree with Thomas, not the naming of a SBP, but the naming of biologicals. And we need to start now because the numbers are relatively small to avoid having many epoietins with many different names in many different regulatory settings. And the WHO has a program, INN program, has a clear mandate on this. The current challenges are enormous because the substances are complex. The number induced the difficulty. Just to give you an idea, we have named more than 300 monoclonal antibodies, and the alphabet is, in Latin alphabet, 24 letters, so we risk to create names that are liable to create confusion with other names. This is a challenge, a stupid one, but an important one. The emerging of new types of substances, we have to invent new policies daily. And the identifier, this is another challenge for an SBP, international set of standards, there is still debate. WHO moves slowly. We have to move creating a consensus, building a consensus. We cannot impose any decision to anybody. But I think that in this direction, Professor Shashkova, correct me if I said something wrong, but I think that this is the direction the group is going to. And I would like to thank you. I hope this is correctly written in Russian. I speak no Russian at all, so I apologize. These three addresses, just at the end, are our extranet, the INN program email. You can write to us any question, anything you need. And the other one is our website, where you can from where you can download publications and lists. Thank you very much.
Good afternoon, dear colleagues. I thank the organizers for the chance to speak here at the conference, and I'll go away from regulatory issues into commercial fields, into figures that reflect modern situation in the market. I will speak about the market of biologics, but I will focus predominantly on biosimilars. It's a new, relatively new concept within the market of biologics, but although a biological product, but different from the product that the medical community, principal stakeholders are habituated to use in their practice to procure for their patients. That is why such a name of my presentation, War and Peace, failed expectations, but still hopes for the future. Thus, IMS company traces sales of medicinal drug, pharmaceuticals, and also evaluates prospects for the development of pharmaceutical market, of its elements, components, and follows the expert opinion as well, and see what were the expectations of the development of biosimilar market just in 2007, the moment they emerged in the market as absolutely innovative products. It was expected that by 2011 the market of biosimilars will reach $16 billion, and nevertheless, we see that in 2 years after the cut-off period, the market of biosimilar is not greater than only $600 million, which was much less than we really expected. But nevertheless, experts insist that the future of the market, biologics market, belongs predominantly to biosimilars, heterogeneous biosimilars, and that by 2020 the market will cost as much as $25 billion, so bring us to the moon. For the Americans is the highest achievement, accomplishment, as you understand. So why there are so many complexities with forecasting, with identifying the specific niche, why there are so many uncertainties about the biosimilars? Let's look first of all at the current situation. First of all, I would say that the regulatory frame is very heterogeneous internationally, and if we forecast positive development because our attitude is positive towards biosimilars, then the United States and Japan are the most advanced in their attempts to organize the market and also in a regulatory frame. They already identified all necessary regulatory acts. They are also supported by payers and biosimilars, and also supported by the physicians, medical community, because they do recognize biosimilars as the drugs that their patients need. On the other side, there's also growing recognitions from the patients, patient organizations. There are some also business stories, such as return on investments into biosimilars. Although biosimilars are not pure innovative products, they do not require such huge investments as usually big pharma does. But nevertheless, the success is required to support this trend. And of course, as for tomorrow, for the nearest future, there are multiple factors that would precondition the achievement of the value of biosimilar markets. equal to $25 billion. There's nothing new in economy. There are two-sided movements from the side of demand and supply. In the segment of biosimilar, we observe some impact from both sides. From demand side, there is a pressure in the advanced developed markets where state is looking for saving more and more money for pharmaceutical drugs. So biosimilars is the solution in such a situation. At the same time, in countries with emerging pharmaceutical markets such as Russia and also China, India, Mexico, Brazil and others, these countries or markets view biosimilars as product that would fill in empty segments of pharmaceutical market. Because there are no modern drugs available or access is an issue to modern treatment, so there is evident potential. There's also support from the companies that invest into these new technologies, and also pressure due to influx of capital, finances into this segment, and also the role of technological companies, leaders in IT, companies such as Fujitsu, Samsung, LG, and some other also want to participate. That also hits up the situation and does not allow Big Pharma to relax. Big Pharma, who are the conductors in the market, it doesn't want to give away their slice of the spice. If we look at the geography of biosimilars, then the most important trend is higher growth tempos demonstrated by these particular sectors compared to the rest of pharma, despite the falling, decelerating of growing, because the world was heading towards the crisis, finally arrived to stagnation. If we look at the geography of this growth, then the United States are the leader in the sales, and they also secure these growth tempers in the biologicals. Well, emerging markets, despite low proportion in sales, they also demonstrate rapid growth. It means that emerging markets are the fastest in their growth in biologic segment. Now about the use of biosimilars. Actually, we divide the international market into three clusters. The first cluster is the market of the USA. Here you see quite a huge and advanced market of biologics. Well, penetration of biosimilars is not so evident. Same situation in other advanced markets, developed markets, Japan, Canada, leading European markets as well. And the third cluster is the emerging markets, where the market of biologic is relatively small, with the exclusion of China. If you see, look at the diagram, you can see that, but for penetration of biologics, then biosimilar are the leaders, and China takes the first place among other countries as related to the depth of penetration of biosimilars. As I mentioned already, one of the most important positions to evaluate current situation of the biosimilar markets and for the perspective is the regulatory component, whether it's in place, whether everything is identified, marketing authorization of biosimilars, market circulation, etc., etc. So, you see here the maturity of regulatory component on the graph. You see that developed economies are much ahead of developing countries as related to comprehensiveness of regulation. No white spots, regulations covering all stages of drug development. India and China closer to the left part of this graph, where not issues are covered by regulations. At the same time, many unanswered questions, and one of them is interchangeability of biosimilars and regulation of this interchangeability. The United States is the largest market, is a benchmark for other pharmaceutical markets, actually formulated all the regulations as it was declared. I won't go into details and conclusions, and nevertheless, circulation of biosimilars is possible only after clinical studies are performed, and it's possible when there are differences in delivery mechanisms in similar drugs. So this is the approach in the United States to biosimilars. Let's go back to Europe and analyze penetration of biosimilars into European markets. So you see the graph as the seaweed in water stream. Thus you can see that the market of biosimilars emerged in 2007 or 8, and it differs depending on the country, type of country. You see here that in poor economies in crisis, such as Greece, biosimilars actually started very actively and grow as well actively. And in this market, biosimilars replace innovator original drugs, so Greece is the fastest. Same situation is in Romania, which is not a key emerging market, but just typical Eastern European developing market, with average attractiveness for investors, not really rich markets. Here you see very fast development of biosimilars markets. In developed segments, we can see more sustained development, some trends, stepwise trends of growth with some cultural limitations or opposition, as you can see on this slide, such as in Spain, Italy, and Belgium. Very fast penetration at the launch stage, but then with new regulatory acts, some such as in Germany, this penetration, rapid penetration, was curbed
So you see geographical differences in penetration, no homogeneity. Unfortunately, I don't have the slides that I was willing to comment, but in a few words, there are differences in therapeutic areas. So, depending on the specific therapeutic field, the biosimilar positions would differ. We can operate different definitions. Some business definitions such as commodity differentiation and biosimilars, depending on the type of the market, can be classified as a commodity where you need a product, rather a product of required price in place, available, etc. So prices of the main importance. Differentiated is just the brand name. There are multiple products, but never the same. The brand is important. Here, for example, filgrastim would be viewed as a typical commodity case with deep penetration in all European markets, with repeat maturation of the market. 80% of the market is filled with biosimilars of filgrastim, and its fate is actually identified by the payers. Different situation with somatotropin, with very modest penetration. It's an example of differentiation where brand is of importance both to physicians and patients as well, and finally payers as well. Such situation is usually associated with chronic diseases when patients need continuous long-term use of drugs. Among therapeutic fields where we anticipate a great expansion of biosimilars in the nearest future, we can identify the largest segments of biologics such as insulins, glucose lowering drugs, monoclonal antibodies, MABs, an abbreviation which is habitual. These are the leading antineoplastic leading drugs in our markets. From the point of view the markets, there is always a key issue for generics. When the patent is expired, there is a fortune for generics. Biosimilars are not generics. We should not confuse, but those who develop biosimilars know this table perfectly, because this is the potential in the market within the nearest future for upcoming decade. $67 billion that are covered by these 12 products with expiring patents. The issue is whether this potential will be reserved for biosimilars or not. Let me illustrate what I say. In biosimilar pipelines, the majority belongs to monoclonal antibodies that are used widely in oncology. Here, science and business has advanced really greatly. You see two first lines. There are numerous drugs, although there are very few in the third phase clinical trials, so we have still to wait new biosimilars. At the same time,
It is becoming quite hot. In this sector, players are elbowing each other. Here we have innovative companies as players, some generic companies that are trying to compensate their losses in the price wars and advanced markets, and they are getting involved into the same war in the emerging market, and there are players from non-profile segments. And those listed below, there may be of great importance, greater than pharmaceutical companies, companies that are involved in contract studies, so they want to expand their activities with biosimilars, expand their portfolio. Quite recently, they performed only different phases of clinical studies, but as for today, they provide new products in collaboration with pharmaceutical companies because competence is an important market component, allowing to perform and be commercially successful. This slide is quite complex, but in a couple of words, you see here how companies, holders of original products, can actually withstand all threats of biosimilars. First of all, of course, there should be a wish to use a particular drug, and also a preparedness of the company to provide lower prices and discounts for the product that they promote. So these are the approaches that would allow companies to keep the original products in the market. It is commerce, of course, but it allows us to progress, not to stagnate. And here you see that biosimilars pose threats to the markets of original biologics, innovative biologics, but they are also under threat. And this situation does not allow us to make clear focus for particular segments of the market, because we observe new generation, emerging generation of biosimilars with different potentials. Some of them will be obviously successful, some are in the area of uncertainty, but they will compete with the drugs that already exist in the market, and this is going to happen within the forthcoming decades. So financial forecasts are really difficult to make. There are specific financial models, but we really fail to provide them by today. So, from commercial point of view, there are still so many uncertainties when we speak about biosimilars, whether they will be commodities or differentiated products. The answer is in the field of uncertainty, and that depends on the therapeutic field where these biosimilars will be positioned, and key issue and headache for the payers, for authorities that identify a reimbursement policy. The main headache is instability of this segment, which is difficult to forecast, situation difficult to forecast, which was actually declared at the very beginning. Few more slides to characterize the Russian markets of biologics. As everywhere in the world, the Russian market is growing faster than the general pharmaceutical market, but biologics is just a small segment of the whole market. The major inputs into the market is the reimbursement drug lists, 7 nosologists. There is some role of the retail market and hospital market, almost similar inputs. In this segment, we specifically outline the Roche company occupying 90%, which is 1/5 of the market, and in 2012, the company kept growing despite all difficulties. The second company is Teva, with the only original drug Capoxone, also demonstrates very good results during last few years, but we do realize the situation can change with the introduction of regulation, with the expansion of biosimilars, if it will happen. And to conclude, the traditional slide from IMS, the forecasts for the nearest future, not speaking of drivers or biosimilars. I want to state that with the decelerated growth tempers, nevertheless, the growth of biosimilars will be plus 10 to 12% annually, which is a positive trend. Thank you so much for your attention. I want to apologize because I need to leave now. So there's an immediate question from the moderator. So what actually you operated, speaking about the biosimilar market? Because we heard a question from the journalist yesterday. The question about the number of biosimilars that got marketing authorization in different countries. If I am correct, in your presentation, you viewed biosimilar as an original innovative drug or not? Because similarity still has to be proved. So how actually you analyzed our markets of biosimilars? If to follow your data, then only in monoclonal antibodies we have 183 new drugs. There's no other country in the world having similar numbers. In the United States, only 2 drugs are marketed. In Europe, 14 only. So, where from did you take these figures? Sorry, I'm not really ready to answer and to give you all details. So it was a broader view or overview. This is also because of heterogeneity of our regulations in different countries. So we had 2 options with my colleagues, whether to follow American standards or take a broader overview. Hopefully I managed.
Okay, thank you all again for having me here. This presentation will be about the traceability of biologicals in spontaneous reporting systems, and I will actually present to you some of my study results, which I will be published in Drug Safety next month. Go to this again. Well, the traceability is not unique to the pharmaceutical industry, to the biological industry, but also a very important aspect of many other industries, as for example the aviation industry, the food safety industry, and also the medical device industry. And well, it's actually the feasibility to link the determinant with the outcome. So as has been described before during this workshop, there are actually 2 major challenges currently in traceability of biologicals. At first, we need to be able to monitor any impact of manufacturing changes for products that have been marketed already. And to be able to monitor these changes, we need to have detailed exposure information up to the batch level of the product. The second major challenge is that we need to be able to monitor the safety profile of biosimilars. It's therefore very important that we can trace back to, well, the particular biosimilar or reference biological product involved. In Europe, we have currently 7 biosimilars approved, well, sold under 14 product names. And among these 7 products, 6 contain the same INN as the innovator. So this might potentially hamper the traceability of well, these biosimilars on the market. So we can say, well, that the usefulness of our pharmacovigilance data sources depends on part on the ability to trace back batches and products when there are biosimilars approved within the data. So we have, for our pharmacovigilance data sources, there are several sources available. At first, you have the spontaneous reporting systems in which, well, the reports of voluntary ADR reports are collected. The second source is population-based databases like CPRD in the UK, in which information from routine healthcare practice is collected. Third, we have the disease registries, as for example, ARTIS registry, in which patients treated with TNF-alpha inhibitors are followed. There are also post-marketing safety studies may also contribute pharmacovigilance data, and so on. Among these data sources, spontaneous reporting systems have historically been regarded as the cornerstone as pharmacovigilance. Because they are relatively easy to establish and relatively cheap. And as previously pointed out, well, virtually all countries in the world do have some kind of system for collecting voluntary reports of adverse drug reactions. Moreover, spontaneous reporting systems have shown by multiple studies to be the main data source for safety-related regulatory action. This recent paper in drug safety, which you can see below, showed that for 55% of the regulatory action, urgent regulatory action for biologicals, spontaneous report was the sole source cited. So urgent variations are direct communication to the health professionals, or suspension or revocation of the marketing authorizations. As we all know the importance of the traceability of biologicals, but we do not have any information available yet on the current status of traceability of biologicals. We conducted the current study. We therefore collected ADR reports from 2 major spontaneous reporting systems, which are the Adverse Event Reporting System in the United States and Eurovigilance in Europe. We collected ADR reports over a 7-year period from 2004 to 2010. From these ADR reports, we extracted a range of data, well, the name of medicinal product, the batch number involved, the name of the marketing authorization holder, type of reporter, roll code of the drug, and year of reporting. And well, the figure below shows that we undertook some efforts to limit any influence of duplicate reporting or follow-up reporting. Our final unit of analysis is actually one product, part of a suspected ADR report administered to a patient at a particular point of time. So it could be that there might be multiple medicinal products reported in one ADR report, and those will all were all analyzed separately. We classify the medicinal products into small molecules and biologicals using the ATC classification system, and roll codes into suspect and non-suspected. And our 2 outcomes were actually batch traceability. Therefore, we aggregated all data in the batch number fields and reviewed the data to confirm the availability. And our second outcome was biosimilar traceability. So as I mentioned before, of the 7 biosimilars approved, 6 contain the same INN as the innovator, meaning that, well, these can only be identifiable upon the trade name or either the name, the INN plus the name of the manufacturer. The exception of this is epoetin zeta, which has a distinct INN from the innovator. Onto the results. So, we retrieved over 2 million case safety reports or ADR reports from both Vigilance and the Adverse Event Reporting System. And within these reports, over 6 million drugs were reported. We identified a total of 590,000 biological in the Adverse Event Reporting System and approximately 440,000 biologicals in Vigilance. For our first outcome, the batch traceability, this table shows the results for the batch traceability. And as you can see on the second row, for the suspected biologicals in the Adverse Event Reporting System, 24% contained a batch number, whereas for 21% in Vigilance contained a batch number. And as you can see on the fourth row, the other highlighted row, that the numbers were substantially higher for biologicals than for small molecules. In Vigilance, only for 3.6% of the small molecules a batch number was available. We observed some variation in the reporting of batches for biologicals. Consumers were actually most likely to report the batch information. For over 40% of the biologicals in Vigilance reported by consumers contained a batch number. Physicians, on the other hand, reported only a batch number for 7% of the biologicals. And well, this pattern actually differed according to the drug class of the biological. For example, the monoclonal antibodies in oncology, batch numbers were available for only 4 to 8% of the biologicals. Well, this shows the time trend in the traceability of the batches. The black line is the US, the Adverse Event Reporting System, and the blue line, Europe. So you can see over time there's not really a sign of an improvement. Then to the results of the biosimilar traceability. We identified a total of 13,719 biologicals, for which at the time of reporting, a biosimilar was approved in Europe. The table below shows the product identifiability for these biologicals. And as you can see on the top bottom, or below, for 96% of the suspected biologicals, well, the biosimilar or reference innovator involved was clearly identifiable. For epoetin alpha, the percentage was even higher, almost 99%. Well, looking into the way that the product was identifiable, the most products were directly identifiable upon trade names. So, for almost 99%, accounted for this traceability, for this identifiability. 0.5% was identifiable upon INN plus name of the manufacturer, and 90 products were reported as epoetin zeta. Before I go to my conclusion slide, I want to point out, well, yeah, that spontaneous reporting systems are not the only source where pharmacovigilance data is captured. We do not know how the traceability is ensured, for example, in disease registries or population-based databases. And but spontaneous.
И система спонтанного отчетности, так сказать, тоже была не совсем четной. Поэтому для завершения я хотел бы сказать, что главная задача – это убедиться, что один из 4, так сказать, тех биологических препаратов, который отслеживался бы в Соединенных Штатах, и это говорит о том, что это важно для потребителей и должно быть отражено в отчетах.
Possible solutions have already been described during this conference. To the use of two-dimensional barcoding and currencing patients and health professionals in the product information, but also making it possible to report ADRs electronically. My final slide, well, we found that the product traceability of biosimilars was fairly well maintained in Europe. It was over 96% across the 3 product classes for which biosimilars have now been approved. Especially for Epoetin alpha, we found that 99% almost was identifiable. This might be caused by the fact that, well, health professionals are encouraged in the product information to record the trade name of the administered product clearly in the patient file. And another reason for the results that we found might be that there is reluctance for substitution, and it's therefore more easy to remember which product was given to the patient. With that, I would like to conclude, and thank you all very much for your attention.
Actually, you have already 4 years of experience with biosimilars in the European Union now. So, I mean, you have extensive pharmacovigilance program, as we see. So, are there any differences in safety profiles of biosimilars versus reference products identified during these 4 years?
Not that we are aware of, no.
And another question, quick question. Are you aware of any differences in safety profile during clinical studies of those biosimilars that are either approved in the European Union or under registration procedure?
Do you mean during clinical development?
Right.
I don't know really.
Okay. So it seems that if a biosimilar, I mean, is properly manufactured and its physicochemical characteristics are shown to be comparable according to European guidelines, it seems that the probability of any, I mean, unexpected adverse events or difference in safety profile is really very low. Isn't it so?
Yeah, still, yeah, that's certainly true. That's the 4 years experience that we have gained so far, although the uptake has not been really high. Indeed shows there's no difference in the safety profile so far, but I think we still need to remain vigilant and, well, really ensure the traceability of these biosimilars on the market.
Another question is both to Niels and Rafaela. So in the report of, in the presentation of Niels, there was, I mean, a clear demonstration that actually any biosimilar can be easily identified in the pharmacovigilance system if the healthcare provider or whoever, I mean, informs about adverse events, reports not only INN but also either the name of manufacturer or trade name. Isn't so?
Yeah, it's true. Yeah.
So it seems that there is no need actually for separate INNs, isn't it?
Well, that's the case for Europe, but I cannot say anything about the situation in other countries. Yeah.
I think he said something very important. Regulatory settings are not all the same, and pharmacovigilance system in place is not all the same. We have a case of 49 fatalities due to Epoetin alpha in Thailand, and it is not possible to know if it is the original substance, if it is a biosimilar, if it is a batch problem, or what it is. But 49 persons died. Do we have to wait until we have the first dead person in Europe?
Okay,
And then just another comment. Professor Vermeer said that 6 out of 7 of these biosimilars were identified using INN. One of these 6 is an Epoetin alpha, which perhaps should have come to the WHO, would not have been named Epoetin alpha, because according to the EPAR, the report of EMA, the glycosylation pattern of this Epoetin alpha is different. And as a result, when it went to Australia, the interpretation of the same guidelines, because the Australians used the Europeans, were so different that they have named it Lambda. This is only 2 cases. And then we have the apoietin zeta that perhaps could, I don't know, because, that has received a different name because they voluntarily applied to us. This is only one example, but the world is global. And I think that it is really needed not to change the INN system, not to invent something different, but to allow to identify what is what. Because if you go from Europe to Thailand and you want to be sure that you are assuming exactly the same substance, how do you do? The trade name, it's important, but it's changing. There is, unfortunately or fortunately, I don't know, not a global trade name. So, and the world is global. So I really think that I don't want to favor one company against another. That is not in the mandate of WHO. The mandate of WHO INN program is only to allow clear identification. And what we are discussing as a proposal, still it is a proposal, is just to allow this clear identification that then will be offered to regulators as a tool that they can use it or not. When we discussed with regulators, I didn't see anybody saying, oh, we don't want. And I think that, on the contrary, the situation is evolving. Because when we start discussing this, some Europeans were saying that they wanted one name for the group. So, apoietin, full stop. And then the situation has evolved. We are not defending market of one against the other. But we are just, because this is not WHO role, so although we are in IFPMA context, I don't think this is what I wanted to say. I was just trying to say we want to have a clear identification. And I think that, we will hear about interchangeability this afternoon, but so far I haven't heard any regulator saying that biosimilars or SBP should be freely interchanged, freely substituted. I haven't heard anybody, because there are some concern. And the perspective of the INN program is maybe a very conservative one. But we name products at the very early stage of the development. We know zero, practically. What do? We want to prevent possible mistakes to be done later on. We don't want to prevent somebody to use an apoietin alpha different from the originator one at all. We want just to allow the patient to know which one is taking of the SBP one, because by definitions they are not the same. These are micro-heterogeneous mixture of substances, and they are not exactly the same.
Well, but you should agree that the problem is not in biosimilars itself, but in a regulatory environment where these biosimilars are approved.
I'm not putting any judgment against, but I agree. There is no bad judgment against biosimilar, and this is not the purpose and neither the mandate of WHO INN program. We just want to allow a clear identification of even the biosimilar. And we, in our, perhaps I shouldn't go so far, but in our discussion there has been even the proposal that we should do it in conjunction with regulators, where there is a comparability exercise in place, where there are guidelines, and then that if we decide to go and having this identifier, this would also identify, perhaps this, I don't know if I have the right to say, but good SBP from bio-questionable SBP that are on the market. And perhaps then we have to do also something for the bio-questionable, but we want to start step by step.
I just want to add comments to your first question, to the Dr. Ivanov question about the biosimilarity in Europe and in terms of safety comparison between biosimilars available. So for 14 products approved so far in Europe, we can say that biosimilarity philosophy of Europe was established very well, because we approved high quality, high similar products with very similar safety attributes, because if otherwise we have seen some differences, they wouldn't have been on the market. So there are some numerical changes, differences between some adverse events which are not deemed to be clinically meaningful. So for that reason, they're highly similar products, and that results in what we have seen from the study demonstrated in this presentation. However, I can't comment on pending biosimilar applications and for those which have been withdrawn from EU submissions.
Just one more comment on the Roman Ivanov first questions. As we heard, there is no serious, unexpected difference between biosimilars and original drugs. But I would like to remind you that we are facing the situation that the more complicated molecules are coming to the market. And we have to be sure that these new products also would be very safe. It's the simple molecules which we used before, well, it looks like the situation more or less clear.
First, I want to thank organizing committee to invite me for giving this presentation, and also all the efforts they made to secure a visa for me to come on time. So first I want to say, this is the first time for me to come to Russia and Moscow. However, Moscow seems is far away from where I live at this moment. But in memory, I think it's very close, because my father visited Moscow more than 50 years ago as a professor. So Russian is the first foreign language I learned as a student. So I still remember a couple of words, Yavchnik and Tavalish. However, Tavalish is not the popular word anymore, so it's cut my language capacity 50% off. So nobody really imagined that if 20 years ago that nobody will imagine that Tavalish is not going to be popular anymore. And just like, at the time, nobody going to imagine that we are talking about biosimilar. And also interchangeability becomes a big issue. Where is the controller? So this, this is about the highlights of my talk today, and the biologics are different from pharmaceuticals. I think we need some background about that. Biosimilars are now generics, and I'm also going to talk about Health Canada's position on interchangeabilities and also global approach for interchangeability issues. So I think this slide illustrates that the biologics are complex, large molecules. And people talking about biologics, they compare biologics with pharmaceuticals like comparing a jet with a bicycle in terms of structure and functionality. So that's how complex they are. So during the last 2 days, you probably have heard enough about the differences between biologics and pharmaceuticals. So from quality perspectives, they are different. And also from clinical perspectives, they are different as well. So these differences make the safety, efficacy, and the quality profiles of biologics are different. So therefore come to the regulation for biosimilars, and has been widely accepted that by regulators and by industry that nobody can make identical copies of biologics or biosimilars and as compared to innovator products. So these differences requires different regulatory approach considerations and guidance tailored to biosimilars. So many countries and WHOs have published guidance document on how to regulate biosimilars. In Canada, biosimilars is also called subsequent entry biologics, are regulated as new drugs by comparison with a reference product previously authorized and marketed in Canada. So it's kind of a hybrid pathways between new drug and a generic. However, we did not change our regulations. We regulate within the existing regulation. So, as I said, why interchangeability becomes a big issue now? We never had a problem with generics, but we have to compare
Нам нужно
between the two. For generics, as we know, drug substance is identical to the reference. So we talk about pharmaceutical equivalence. The only clinical study required is pharmacokinetic study, a comparative pharmacokinetic study. If pharmacokinetic equivalence is established, then therapeutic equivalence is assumed. So therefore, all indications that a reference has will be automatically extrapolated. And then at a different level, automatic interchangeability or substitution will occur. So, but when you look at biosimilars, because drug substance only similar to reference, there are many more steps and requirements for biosimilars to establish similarity as compared to generics. So there is no, the indication is no automatically, the efficacy and the safety is only comparable, is not, equivalent. So indication extrapolation will be case by case. So that means a biosimilar product may not receive all indications that a reference will have. And interchangeability generally is a no. It's not recommended in Canada. Internationally, at this moment, only FDA has, their legislation gave FDA the authority to declare interchangeability, but they still haven't decided what to do yet. It's a very challenging target. So when we said the interchangeability is no, I think we base on some scientific concerns, as we have discussed in past 2 days. I think first is a quality concern. The quality you cannot, from quality perspective, you cannot make two products identical. So they are not the same, they're only similar. So the similarity profile may also means they have different safety concerns. Any manufacturing change may have significant clinical and safety consequence. So talking about the safety, because besides quality concerns, these drugs are complex, so they may have different impurity profile. So again, the consequence, if you grant automatic substitution, then the safety may not be the same. The biggest concern will be immunogenicity. So I think we have two concerns. One is that during clinical development, because we are talking about a reduced clinical package. So it depends on which study population. That population may not be very sensitive to detect all immunogenicity effect. So I guess Thomas talked several times, you may have to do different efficacy and immunogenicity testing among different populations. We do have that concerns when when company come to us. The case is for the rheumatoid arthritis treatment with infliximab. So they may use that indication. Infliximab is with methotrexate. Methotrexate is immunosuppressant. So that population will not give you the best immunogenicity profile. I think there is publications demonstrate that. So we have recommend to biosimilar manufacturers, you may be better off to do a comparison in a monotherapy indication, or at least you may do both, monotherapy and a combination therapy. Another concern is that, as I have said, all indications may not be granted to biosimilar product. So if you do automatic substitution, then you may use that biosimilar for indication that not be authorized. So the last one is about traceability. I think people have talked about it. I'm not going to say too much about it. It's just in case that in some countries that you don't have a good system, then you cannot really track, what's the causality at the individual patient level, and from market perspective, you cannot trace the product that caused the problem, so you can remove that from the market. However, this in some countries, not in all. Well, this slide shows you that in the future, you have many companies to do biosimilars, including some big pharma and also big biotech company. So you can see that there is a new kid on the block, Samsung, which is, famous electronic company. So we are kidding. If they are going to add a remote control to their biosimilar, so you can turn on and turn off their product, which seems better, but is not going to be biosimilar, because biosimilar means you have to be as good or as bad, as a reference. So Health Canada does not declare interchangeability neither for pharmaceuticals nor for biosimilars, because it's not within our authority. It is in Canada, is at provincial level. And also it's up to physician to make decision which product they're going to use to treat patient. So they have a choice. However, we don't have authority, but doesn't mean we don't have a position. So Health Canada does have a position. We do not recommend automatic substitution. So we indicate that biosimilar is not a generic. It does not, authorize a biosimilar doesn't mean they have therapeutic equivalence. And also due to manufacturing change, that Health Canada does not support automatic substitution and to the reference product. So that's our current position. I also want to explain a little bit about interchangeability and substitutability. In Canada, it's two different things. I think internationally, more or less, it's the same, but depends on how the definition in each country. So interchangeability in Canada means that you substitute the product to a lower cost generic. And the condition is that there must be bioequivalent. That's the term is used for generic, is not for biosimilar. As I said, biosimilar, they are similar, not equivalent. So that does not apply to interchangeability at a pharmacy level. So and also in Canada, you want to interchange. they should have a committee to, assess that equivalence, and they have a list. So pharmacists will look at the list so they can automatically substitute. So if the drug is not on the list, they are not allowed to substitute. Substitutability, in Canada means, or therapeutic substitution means a different product. However, it has functionally, equivalent to the prescribed drug. So this is a different situation. It means a different drug. So which is much like the biosimilar will fall into this category. But this is a medical decision. The pharmacist cannot, do automatic substitution. Physician has the practice of medicine is, will be decided by a physician that how you're going to use the drug. So what we can see that if you have a new patient you need to treat, the physician can decide, he or she can use either biosimilar or originator's drug, because when biosimilar authorized in Canada, it's become a standalone. So if it's a growth hormone, it's going to be a growth hormone. So the physician can decide which drug to be used. However, it's not going to be interchanged. So that means if you already start a patient with a biologic, you don't want to switch forth and back. This doesn't mean only apply to biosimilars, it's also applied to all biologics. I don't think it's a general practice they do this switching. Okay. Unlike generic drugs, you go to a pharmacy, they say, okay, this time we have manufacturer A for this one, next time is B, next time is C. So they can change all the time. But for biosimilars and for any biologics, I don't think they do that. I think that some terminology is very confusing. Okay, don't look at the definition. It's just what I try to emphasize. If you really want to, for a biosimilar company, say, I want to claim my drug can be interchanged, then as a minimum, we say you should demonstrate that in clinical studies. That we recommend a 3-arm parallel design study. So first, you treat a biologic of the choice, either similar or the reference, and you demonstrate the safety, efficacy first, and then at a time point, you can switch. Then you follow it for a longer period to see if there is any changes in safety, efficacy, and immunogenicity. I think some people recommend a 2-arm study, switch over. But we believe the 3-arm is the best because you have the arm will keep treating with reference, which is going to give you a better understanding of the safety and the efficacy. So I want to mention a little bit about international. I think a different speaker has talked about it. So in Canada, as I said, we have a position, but decision is at provincial level. However, at this time, the 10 provinces and 2 territories in Canada, 9 of them has declared biosimilar is interchangeable. So in the EU, the decision is at each member state. And at this moment, what I can find from the internet is about 15 nations have said biosimilars are not interchangeable. So other countries within EU, I'm not sure, what their position, so someone can update on that. Well, with FDA, that their legislation has given them the power to declare interchangeability. However, it's a very challenging task. I have been talking to FDA colleagues, And at this moment, they don't want to say anything about it. However, we did have one FDA DIA biosimilar conference, and some discussion, were made at that conference. However, when you look at US legislation, they said, the definition for interchangeability or interchangeable means three things. First, the drug's product is similar to the reference is number one. Number two, when you give the product, you expect the same clinical results as reference in any given patient. And also the safety profile is pretty much the same when you switch, like you gave that reference continuously at an individual level. I think there are some legal interpretation is at individual level. Or it really means you have to demonstrate in actually each patient, or is the collective results. Biosimilar company said no, it doesn't mean individual individual patient. The brand name said yes, it seems required that at individual level, which is a very high bar, and it's not easy to achieve if that's really at individual level. So that's why US FDA has to come up some solution, how to, from a scientific perspective, from clinical perspective, how to declare interchangeability. Well, some of their states went ahead and they said they have, I think at their states level, they have very similar laws, similar to Canadian at the provincial level, that you have to be equivalent between the two products, then you can declare interchangeability. So they try to amend their law and saying that if FDA decide which one is interchangeable and the physician does not require brand name, they can interchange. So in conclusion, biosimilar is not identical to the reference biologic. I think that we have to keep in mind they are not the same. So each country has its own approach to interchangeability of biosimilars based on the national or the local laws. So in many countries, automatic substitution of biosimilars is not recommended. So that's the end of my presentation. I guess I still remember that, suddenly I forgot how to say it. Specible?
Thank you for this introduction, and I'm sorry I lost the last few sentences of the translation, but I understood the rest, so thank you very much. And thank you also for the invitation to speak on IFPMA, AIPMN, and the Russian government. It's really a pleasure to be here. And we talked about energy this morning. I'm based in Chicago, so for me the day is just starting. So if you can see me jumping around on stage, that's because my energy is just here. But I am aware that I am almost the last one between you and dinner, drinks, whatever you prefer. And the aspects, talking about regulatory and medical aspects of interchangeability of biosimilars. We just had a talk on the regulatory aspects. We had a very good talk also on the clinical aspects, so you can wonder what possibly new can this guy tell me, which we haven't heard already. But I will hope to live up to that expectation, because I think I've got a couple of points to share that I'd really like to point out. And starting out by having heard the discussion, and I'm really sorry I couldn't be here yesterday because I was speaking in another historic European capital, Berlin, yesterday. And I spoke about the quality considerations for biologics and biosimilars. And one question during the Q&A session there was, aren't we overdoing what we are doing for the simple biologics? And I actually paraphrased and I returned the question and I said, aren't we taking the concept of biosimilarity too far in the case of antibodies? And that's exactly, I think, the fine line that we are walking here between what to understand biologicals, biosimilars, what are complex molecules. The discussion went further and that's really a point I'd like to make, not only as an AbbVie representative, but also as a person well experienced with biologics, I would say over the last decade or so. We are talking about molecules that have been on the market for 6 years. 7 of those have been approved in Europe, not more. We have 14 products, but 7 unique biosimilars. 2 in Japan, a couple of others in other jurisdictions. There's really not that much experience yet. And on top of that, I would say whether or not issues have arisen, I think going forward, it's actually very important to let common sense and science guide the principles that we really need to apply going forward. And I always take the example of learning how to ride a bike, which you can give to a kid and learning to give in a couple of days, just sending them on the road. It does probably take a couple of weeks or months, depending on the country you live in, to actually get a license to drive a car. And very few people are actually allowed to fly a plane. And with that comes increasing responsibility and increasing complexity. So that's what my introduction is. And I have a couple of slides, which I'm sure you have seen in very many other ways as well. I have my version of it. This shows the complexity of molecules of biologics. Most of my talk will actually be focused on antibodies, which are roughly 1,000-fold larger in size than the average generic chemically synthesized molecule. And with that increasing size comes a lot of complexity and a lot of concepts, and we've heard that, so I'll quickly go through this. A lot of different domains, different subunits, light chains, heavy chains, disulfide bonds, which are important for the structure and for the stability. And in many cases, and also today, we tend to focus on some of the post-translational modifications, in particular glycosylation, and in IgG1 molecules you have 2 particular sites of this modification, which are exemplified here. They're naturally occurring, but in products they are always different. And they not only are important for the immunogenicity, but also for the, very often for the stability of the CH2 domain. So there are other reasons why this is important. But this is not where it stopped. This is just a small list of heterogeneity and modifications in antibodies. So it's more than just glycosylation. All of this is important. There's N-terminal heterogeneity, C-terminal heterogeneity, the famous C-terminal lysine, splice variant, mutations, oxidations, deamidations. The list goes on, and this is just a grocery list. So that's something that we really have to keep in mind. And then, of course, the stability, physical stability in terms of aggregation, it's mentioned already, fragmentation, but also the stability of the product in terms of concentration, thermodynamics, etc. I guess what I'm trying to say, like many other speakers before me, 20,000 atoms are really a lot to track. And we don't have the detailed structure in many cases. We don't always understand their complexity, their mechanism, even diseases they treat. So in order to understand what's important functionally and to understand what's maybe different functionally, is that you have to test it. And that's really the paradigm of what we're working under. I'll skip these couple of slides because these points have been made. So I've been asked to give some perspective, some clinical regulatory perspective on substitution in particular. And I thought it would be good to define how, or at least to list how we define the different terms, because in many cases they are used in different. So in our case, in our view, interchangeability is really a health or a regulatory authority decision. So this is not a practice by a pharmacist or a doctor, that's a decision by an authority. And what you see in the 2 bullets underneath is so far the only regulatory requirement or standards that have been formulated when it comes about interchangeability. This is from the FDA, and it's been mentioned already a couple of times. And the FDA is the only regulatory authority in the world right now who is charged with finding and defining a standard for interchangeability. No other regulatory authority, to our knowledge, and that was, let's say, as of 5 minutes ago, is actually currently doing that. So we're facing the situation that either standards for interchangeability are going to be defined, and they haven't been defined yet, because what's been written here in these bullet points is sufficient for legislators, but it's not sufficient for scientists and clinicians to figure out what actually is meant here and how that should be proven. And then there are a whole bunch of other countries and jurisdictions that do not touch upon interchangeability and basically said, we are not going to talk about this and we're not going to allow it, or at least certainly not encourage it. Substitution is a pharmacist action which either allows or requires that pharmacists to substitute the product without, often or sometimes without the knowledge of the physician. And of course that happens a lot in the case of generics. And switching, that's what we call switching, is an active decision by the physician. So substitution is, let's say, a practice at the pharmacy. Switching is really a physician, hopefully in consultation with the patient, to really decide on switching treatments. And that could be between a chemical drug and a biologic, between one biologic in the same class to another, and of course in future, and in fact in some cases it's happened already, between a biologic and a biosimilar. That there can be reasons for that. And this has been mentioned. In the case of generics, when pharmaceutical equivalence has been demonstrated, usually that also demonstrates therapeutic equivalence, and regulators have designated them as interchangeable. And then depending on the local or the country legislation, either it's required or allowed to substitute. Now for biosimilars, that's not the case. Pharmaceutical equivalence in this case doesn't mean therapeutic equivalence, and granting a biosimilar status by a regulatory authority, so far, not in any country, has implied interchangeability. So it's biosimilarity, not interchangeability. So therefore, automatic substitution is not allowed. And depending on the regulatory agency, as I mentioned, it may or may have to be shown by the biosimilar sponsor. I thought it would be useful to have a snapshot of a couple of major jurisdictions around the world, how they've treated. And again, I won't repeat too much of what's been said already. I actually have a more detailed slide on the EMA and the FDA. But for example, Australia, Brazil, Australia doesn't apply substitution at this point. It may consider it in the future, but there are no direct plans. In fact, TGA is currently considering revising some of the biosimilar guidelines. In Japan, interchangeability and automatic substitution are highly discouraged. EMA, as mentioned, has no authority on substitution, and the decision is left to member states. But so far, no country in Europe, no country within the EMA jurisdiction, has actually explicitly authorized substitution of biosimilars, or biologics, I should say. Brazil has guidelines since a couple of years, but it doesn't address interchangeability or substitution. Health Canada, that was just mentioned, it doesn't support automatic substitution by way of the position by Health Canada, but allows the provinces to determine. And the US, in a similar way, but very different, FDA is charged by the legislation to define the standards. And then the act of substitution, how that is actually going to happen, that will be defined at the state level. So this is much of what is currently also being discussed in the US, and that's what I would say, been exposed to almost every day, how that's going to work. And I'll come back into a little bit later, so I won't spend too much time on that. As mentioned, EMA, the dossier, so in the case of the marketing authorization application, that does not contain any evidence to substantiate interchangeability. So there's no requirement for a clinical study or anything to show this, simply because the EMA doesn't decide on that. And the legal decision is left to member states. I thought it would also be interesting to quote a very recent document that was put together by the European Commission. It's a consensus information paper which actually was created by, I would say, just about every stakeholder in Europe, industry, European, and everybody, talking about the future of biosimilars. And I'd like to quote one sentence in the report, which says, "It's important to note that biosimilar market uptake has been possible despite the fact that substitution between the biosimilar and its reference medicinal product is not practiced at the pharmacy level." So in Europe, which is the region with by far the most experience with what we call biosimilars, so they are not quite questionables or whatever you call them, in fact has experienced quite significant growth rates. And again, yesterday, I had a very nice discussion with my colleague from Sandoz who showed a 45% growth rate of the biosimilar product in Europe, and all of this happened without substitution. So in other words, it's the marketplace as well as other reasons which may help grow biosimilars for the reasons that have been discussed: affordability, increased access, etc. So the point I guess I'm trying to make is, if you are thinking about substitution and interchangeability, let's not forget that market introduction, simple working market mechanisms are usually sufficient to actually allow this access. So no country has explicitly authorized the substitution of biological products from different manufacturers. And in fact, a number of actually have explicitly prevented that. So far about a little bit touching on the regulatory perspective, a little bit on the clinical perspective. And now I'm going back to the FDA being the only authority that has said something about it. It's mentioned already, it has to be shown. The foundation is it's a biosimilar. And in fact, I think it's interesting to mention that I've been at various conferences where FDA has publicly stated that it's very unlikely that they are going to approve biosimilars in a first pass as interchangeable. In many cases, they have mentioned what they're probably going to look at is a biosimilar, approve it as a biosimilar, look for post-marketing experience in combination with clinical data, and then consider interchangeability. So personally, I think it's unlikely that we will see interchangeable biosimilars in the US for the next couple of years before those standards have been defined and before the first biosimilars have actually been demonstrated to the marketplace. And again, that was also discussed yesterday with regard to market exclusivity. But back to the science. It can be expected that produce the same clinical result as the reference product in any given patient. Now, as a clinician, I thought about this a lot. How on earth am I going to demonstrate that? Because if you look at it in any given patient, same clinical result, very quickly you're looking at huge studies, very complex studies with crossover designs that is going to do that. And of course, there's a legal aspect. What does any given patient mean? The other aspect, in case of multiple dosing, and in many cases, especially with monoclonal antibodies, we are talking about chronic treatments, or sometimes even lifelong treatments. So in terms of the risk, in terms of safety or diminished efficacy of alternating or switching, and very often we focus on the switching part, not very many times it mentioned the alternating part, because alternating actually means that you switch back and forth and back. And the question is that relevant? Whether or not it's relevant or not, this is the legal language that should address this topic. And again, for for chronically treated drugs, I think it is relevant, because for other reasons than medical reasons, patients may be switched between years or periods. If they switch insurance, for example, in the US, they may have different formularies, so they may have different drugs on their list. So for that reason, they may say it. So switching back and forth is actually a reality, in my opinion, in many countries. So what are factors to consider? Well, safety signals during the biosimilar development, if any new signals arises, although that's not extremely likely, but of course possible. Known safety issues with the reference product. Many times discussed immunogenicity risk, the frequency, severity, anti-drug antibodies, and of course the correlation with any clinical outcome. Very important also, and these are aspects that are often discussed in different realms. What about safety and efficacy in different indications? For example, if we are talking about a biosimilar that has performed clinical studies in a number of indications, but not in all of them, and in fact has been approved for all of them. In other words, there are not even clinical data in that indication. Then we're talking about interchangeability in that possible indication. So in other words, we may be building uncertainty on uncertainty, and that's also something to keep in mind, and I'll come back to that. Subpopulations, I think it's been mentioned very well by the previous speaker about the example of a study in methotrexate-treated RA patients, which would not necessarily be very representative for a study, an immune-competent population in other diseases or populations. And of course, the sensitivity of the population to of the drug to minor changes. So these are some of the aspects, and I won't do that. You can talk for hours on this, but some of the aspects that go into the design. So how would that look like? And most of you or some of you must have seen this before. A simple design would be a crossover design that would switch from the biosimilar to the reference product and the other way around. And also should at least have one arm that keeps the patients on the reference product. The question is, should we also have an arm that keeps the patient on the biosimilar product? I just throw it out there. For chronic diseases, and that's an additional complexity, we are talking about naive patients, but I think it's also important to treat patients that are already stable on their current treatment, because that's actually what's going to happen. We're not only talking about patients that are new on therapy and are going to be switched, but are actually going to be switched already on therapy. And that raises a number of questions, both in terms of feasibility and I think in ethics. The other point I'd like to make out is the red dotted arrow, which is actually the washout period. And that's not very often shown as well. Most biologicals have a very long half-life, typically a couple of weeks, sometimes even 2 months. So in order to make a correct determination of the actual effect, you have to wash out the previous biologic from the new treatment. And that very often, in terms of, to do a proper clinical study, means that you actually leave the patient without a couple of weeks of medication. And again, raising questions on feasibility and ethics.
То есть время вывода вот такое, и оно может быть разным до 2 месяцев, переход на другой препарат. Если я хотел бы подумать, что происходит в настоящем в нормальном мире, когда мы рекомендуем переход с одного периода на другой, надо посмотреть, какой период вывода из организма первого препарата перед тем, как мы перейдем на другой. И у нас на рынке могут быть ситуации, когда у нас 2 референтных продукта, может быть 3 или 4 биосимиляра. Возникает вопрос, это говорит о том, что мы можем переходить с одного биоподобного на другой, или надо провести отдельные исследования. Технически, наверное, нужно проводить дополнительные исследования. Проводить мониторинг по одному, по другому. Различное число комбинаций сразу бросается в глаза. Я уже говорил об этом, кстати, эффект переноса вследствие длинного времени вывода из организма, состояние пациентов с течением времени, как они почувствуют себя.
These are typically long studies, so we're dealing with patients whose clinical status may either improve or deteriorate over time. Ethical point, for the patient there is no benefit because switching itself is not known to raise any benefit. So in terms of doing this type of study, I think it's going to be interesting how this is going to be viewed by ethics committees because of that fact. And recruitment issues, as I mentioned. I can imagine that would be quite difficult. Study considerations. It should be sufficient to capture, of course, the equivalent efficacy, the time course of predefined safety events, depending on the product. The size and statistical power should ensure the demonstration of equivalent efficacy, and, of course, also the detection of critical safety events, including loss of efficacy due to, or at least potentially correlated with switching. And again, it always comes back. Immunogenicity comes back in any topic you talk about, biologics or biosimilar, but drug neutralization or loss of efficacy is one of the major risks associated with switching. And you will see a quote, "Lost ground is lost forever." I've spoken to, and I'm talking about dermatologists, rheumatologists. I've spoken to a lot of physicians and asked them about their opinion without saying anything in it. What do you think? And he said, it's just about the last thing I really want to do. Because once I have a patient who is stable on treatment, and I have only 2 choices maybe. Maybe it's a patient who needs that treatment for the rest of their lives. And I have 1 choice. I'm not going to take any risk, no matter how small, to run the risk that I'm risking the loss of efficacy or whatever adverse event for that patient. And that's what many physicians have personally told me. That's the paradigm of what physicians are doing. I'm not a physician myself, but I can sit in their footsteps. I already mentioned it, and these are the last couple of 2 or 3 slides. What about substitution, interchangeability, and extrapolation? can we say that by demonstrating it for one category of patient, very often even a subpopulation of a certain disease, can we really say that it is interchangeable for all the indications? For example, for anti-TNFs that we are by nature quite interested in, we're talking about molecules that have 7, 8, 9 different indications, often with very specific labels, very specific subpopulations. Can you really say that clinical data from one study in one population determine interchangeability in all of these? It's a question. I'm not saying it can, but it's a question. And then you have the possibility, at least the distinct possibility, if that answer is yes or no or maybe, that you may consider the situation where a biosimilar may be considered interchangeable in one indication, but not necessarily in the other. I don't think, I think it's fair to say that might really create a lot of confusion if that's the case. And I don't think any of us would want that type of situation. I'll skip the post-pharmacovigilance. I think this has been discussed extensively already. So I'd like to proceed to the last slide, which is summarizing a couple of points I've been trying to make. Current science does not justify interchangeability or substitution for any biologic, including biosimilars. It's simply not been demonstrated by any study. No study has been done showing this as a primary endpoint. Whether it's a biosimilar, biologics, or any type of those products. Approval of a biosimilar should not be intended to allow interchangeability with their reference product, not on the basis of the requirements that we have discussed today and yesterday, because demonstration of interchangeability requires higher standards and additional clinical data in all indications, or if pertinent patient subpopulations, should be demonstrated. And I think I've hoped to show you a flavor. There's a whole world beneath the discussion on switching studies. They're complex for technical, ethical reasons, and very, very little data is actually available on this topic. And I'd like to show, I'd like to finish with one additional thought. We've really thought hard, and by we I mean a group of scientists, external, internal, whether it's actually possible to demonstrate at all interchangeability, whether it will be possible to adhere to the standards that have been defined by FDA. And for me, that's still an open question. I really am not convinced that it will be able to show this for biologics, especially for the complex biologics and the complex diseases that we're treating. We're working hard on it, we are thinking about it, but I'm not convinced it is. So with this, I'd like to finish and take any questions in the panel, I suppose. And I'm happy that the weather today is like it's shown on the picture. Thank you.
In principle, as my speaker already said, it's actually made my job quite easy in terms of reiterating some terms. So just talking about WHO definitions, which actually given a bit more broader framework, I think, but they're actually very much similar to what already been said, that interchangeability is something which is ascribed by regulatory authority, something which is permitted on a basis of clinical data supporting interchangeability. This clinical data can be of two types. One is clinical data derived from studies comparing the product switches from the reference to biosimilar and vice versa, and also pharmacovigilance data, which might be supporting post-marketing surveillance and lack of safety issues arising from switches occurring between reference to biosimilars and so on. Whether one type of data or another will be sufficient to demonstrate that remains a question in terms of FDA position. But the rest of the regulatory authorities don't have really solid view on what type of clinical data required and whether it's required at all. So as I mentioned, in Europe, we do not require this data as pre-approval. We do not regulate interchangeability itself. So this is left to national authority. Substitution is something which is driven by pharmacists or can also be done by medical practitioners, but without clinical data to support this. And this substitution on the pharmacy level is usually, or theoretically, would be at greater risk because pharmacists not necessarily will contact physician for decision about substitution. And finally, the third level which is automatic substitution, often done for economic reasons, and also driven by policies imposed and dictating prescribers to actually, or pharmacists to actually switch patients from one product to another. And as Madame Maximkina already mentioned, that there are economic and therapeutic aspects to interchangeability and substitution issues for any regions. And I guess from European perspective, we have experience of national authorities managing these issues on a local level. And I have to say that, yes, there might be some automatic switching happening in some countries where this is not prohibited. So there are national authorities which prohibit on the national level this switch, automatic substitution. And there are some countries which allow that. However, we're mindful that at the global level, there might be further issues which might complicate situations. So some sort of level of harmonization will be required in years to come to ensure that we're all working on the same page. And just one more clarification with regards to switching. My previous speaker said that this is actually physician decision about switching between different classes of products, and often between, for example, small molecule drugs to biologics and so on. That term is often confused with switching between biosimilar and reference. So this is very much broader term, which often encompass all three different things which I mentioned: interchangeability, substitution, and automatic substitution. So this is like a broad term, just switching. Hope that clarifies the position.
Thank you. I think it was a very productive discussion, and it's very impressive to see what's been achieved in only 2 days. So 2 days is relatively short period, and we had these time slots of 2 days many times in previous years, but we didn't have such an excellent opportunity. So I think the organizers and the Ministry of Health and the Russian government really took a very important action. And while we are closing the conference now, we are basically opening a new page, and we are really initiating a very important events in the near future. So I believe that these 2 days actually gave everybody an excellent opportunity to learn who is doing what, to learn what we do have today, and also what is missing today. And it's now up to everybody to express opinion, what needs to be put in place and what we would like to see tomorrow when we meet again, and we may feel proud of doing something important and saying, well, what we agreed in Moscow in May, when it was really summertime, not really the spring, did we achieve enough? Did we make good progress? Did we follow on what we agreed to follow? And one of the obvious actions that need to be taken, I think, is some sort of creation of a network for information and knowledge sharing to make sure that everybody who is concerned is actually receiving the appropriate information, and it's receiving that information on time. And that is of critical importance, not only for this region and this area, which is actually part of the European region, but it's also very unique in terms of manufacturing, in terms of regulation, in terms of historical and developments that actually occurred. So one of the questions is, what would be the most effective way to exchange information in future? Would several countries that were represented here be willing to think about starting some sort of networking activities? And then maybe at the later stage, when they are established, would they be willing to share their experience with other networks? And if WHO could actually help in setting this kind of network for information and knowledge sharing that would be of great value for completing the world map, because in previous discussions I mentioned we had lots of activities in other parts of the region and the world, but obviously not here in terms of biotherapeutic. So that would be the first obvious action that we need to think about. And then in terms of further sort of actions, I also wonder whether, in addition, we understood that our excellent organizers, Janice and Chiara, and also the entire organizing committee actually plans to publish presentations, so everybody will be able to see these slides on the website, and to continue building on that, because there was a lot of details. We couldn't really digest everything. We heard a lot, but we still need to work on these data. So in addition to that, would it be valuable addition to have a summary, maybe just a very brief summary, but key findings, key topics discussed, key issues that we actually found in these 2 days, so that we can actually follow on that and act in line with the current situation and needs. And the third point that I would like to mention is that mapping up the expertise that exists. And I'm really impressed. I think we heard really excellent speakers from all over the world and many institutions, but also Russian-speaking experts are excellent. So we have a very good starting point. And mapping the expertise and identifying what is already in place, what is the capacity for handling the regulation of biotherapeutics in general and biosimilars in particular is, I think, important action to be taken. Once we identify what is missing in terms of expertise and capacity, we can then discuss what are the mechanisms to put that in place, to develop this, to offer maybe technical assistance, to explore a possibility of WHO collaborating centers for biological standardization to assist on certain aspects that may be most needed. So I think these 3 points are maybe setting the scene, and I'm sure that my colleagues around the table have a lot more to add. So I would stop here.
Thank you. Alex, would you like to add something?
Thank you very much. Well, first of all, I would like to express my gratitude to organizers for inviting me here. And I think this event was really successful because in 2 days, first of all, very clearly, we understand that Russia moved into a new era of using biological products and biosimilars. And this is not something coming, it's already taken place. The second point I would like to make, that we achieved yesterday and today, that we have discussions from different points of view, sometimes polarized, but one clear thing, that with biosimilar and biologics, all stakeholders need to work together, because it's a very complex and large business in which many different sides will be affected. Patients, regulatory authorities, prescribers, physicians, payers, so all sorts of parties will need to be involved and work together to come on the same page. The third point I would like to say, that we need a lot of education for everyone. We educate ourselves here when we talk about issues with biosimilars and biologics, but different type of education needed for medical students in Russia who will be using these biologic products and biosimilars tomorrow. We need education of patients, what type of risks and benefits they will be exposed to when they will embark on treatment with biological products and biosimilars. And equally, we need to educate other stakeholders, again, payers and so on. And in order to achieve this, as Ivan already mentioned, we need a lot of communications, and that can be achieved by either some sort of press release from this current session, or also some publications which can be done on official level. And I know that Dr. Mironov and Dr. Maxim can already involved a lot in doing and achieving this by publishing various manuals and various papers, and I think that's a very important thing. So communications is very important because that serves a tool for education. Thank you.
Thank you, Alex. Rafaela, would you like be next? Because I highly recommend that Elena Maximkin will conclude from the Russian MOH.
Thank you. I think I am really the outsider, and I want to feel humble in this group. I have to feel humble. So I would like to thank everybody who has told. I think that I am the outsider. I feel humble. I want to feel humble. I want to thank all of those from whom I have learned many things. I would like to thank the participants for being brave, being here, being listening, asking questions. I think I'm not a diplomat, I'm not a bureaucrat, I'm not somebody who is very capable in making very good speeches. But I would like to say that we have, if I have something in my brain, I think that we have to be wise, yes, perhaps conservative, yes, but inventive and creative. So what we have been told is not a way to stop, but I think is a way to start. And not to be against, but to be pro. And just to think the way we can better serve public health and people receiving medicines, but very good medicines. Thank you. And again, thank you for inviting me. It has been a real pleasure to be an outsider in this group.
Thank you, Rafaela. Shinar.
I would like to extend my gratitude to the organizing committee because back in March I was in the United Arab Emirates and the event there was initiated by the Ministry of Health of Emirates under support of the Pharmaceutical Association of Manufacturers. Unfortunately, then I couldn't find a single Russian-speaking person there. There were representatives from the African and Arab countries, and I thought, why don't we have conferences in the territory of the Russian-speaking countries? Yesterday I was lucky when the organizing committee set up this conference, and I'm happy that I took part in this event. And the most interesting is that in this conference we had information from the regulators directly, where the vast regulatory practice is underway. And it's very pleasant to hear that such a person as Alex is working close to us, to whom we can approach and we can speak Russian to him, and he will explain things that we are missing in English. And this is a very good thing to know. And it was good to hear that the company originators of the biologics, they are so willing to share their experience in the area of development of the medicines. And the Sandoz company, as a generic company, is doing its best to be on par with originators and also striving to manufacture bioanalogues, which are having the same quality as the initial medicines. It was interesting to see the approach of the WHO, and we have to move on in this area. We should start working jointly. And we mentioned today about the need to work out unique identifiers to be used in the post-marketing vigilance, in supervision of the medicines. And I think we shouldn't redevelop, reinvent the wheel. We should make use of what the WHO has developed. And the successful EU, so that from this standpoint we are together. We are on the same page. And then I would like to expand on the following. following. Just know that CIS countries, Russian-speaking countries, should develop a unified approach to registration of pharmacovigilance of biologics as a whole, including biosimilars. So we shouldn't develop against something if the leading countries, the EU, have developed. So why should we make our lives difficult? We should stick to the practice that has proven its suitability. And on top of that, today we mentioned, we spoke about interchangeability, about the switching, and this is a very difficult issue, and we understand that there is no substantiation to that we have to replace or to interchange these products. If we are purchasing using the budget money, from an ethical standpoint, I think this is just a way of risking, of putting on the line the life and the health of our patients. And we should be very careful in making these decisions. My proposal to the originating company is if the patent protection term expires, over the period which they compensated their costs to develop, they can speak about bringing down the cost of the medicine when the patent has expired, and then gradually coming down with the price when the analogs are coming to the market. Because otherwise, the chances are higher that either these companies stick with the practice of high prices will be taken out of the members, which is counter-effective. Because the practice is such as, regardless of the fact that there is a biological synthetic medicine, as soon as the first analog is on the market, generic or biosimilar, the company originating stepping down with the price. This is a debatable issue, and we would like to implement a post-marketing survey. So I think we should be consistent. Biological medicines designed to patients which are not big in number. So from this standpoint, we should develop the register of patients that will allow us to keep track of statistics, to calculate the frequency of adverse effects. And of course, it's desired that the WHO and representatives from pharmaceutical companies address regulatory bodies in developed countries to provide us support, to educate us, to give us knowledge in the area of expertise dealing with such medicines, because we cannot say that we are well equipped here. Thank you, Shinar. Elena, you have the privilege to have the final word.
Спасибо, Шинар. Елена Анатольевна, вы имеете привилегию сказать последнее.
I'm quite pleased to have at least some privileges, because in the morning, when I visited my office, I was surprised to hear that in rating, my Ministry of Health was given the lowest score from the standpoint of transparency, with which I disagree, and justification is provided by this very conference. We are taking an active part in participating in setting up the conference. And in fact, this conference happened because the Ministry of Health of the Russian Federation was the culprit, was the problem child. Because how we understand the interchangeability, my colleagues are sitting in the audience because the commission of the government to introduce amendments into the Law 61 dealt only with one thing: to streamline the procedure of registration of medicines. And the draft that everyone is reading through and which is 51 pages long, there are many corrections that are recommended, and the discussion is not by chance that it happened here at this conference. And we see to what extent the market is ready to actively get down to regulating procedures, to detail specific processes. And I am quite grateful to address the organizing committee of the conference. And I think this is not the last conference, that we will have similar conferences in the future. Also, I would like to say a thank you to my colleagues, to the leading organizations spending their time, found the time slot in their schedule to share their experience, knowledge, to give us some things to brood and to think about, to establish the contexts. That said, this is the very beginning of the process, and of course we will make use of this opportunity to continue, and this is the opportunity for us to continue dialogue within the community, inside Russia and CIS. And I think many of those who attend this conference will attend the conference, the forum in St. Petersburg. I have it in my diary and I want to get there and continue our debates, because the competition and the possibility to position Russian pharma in the context of international pharma within the framework that provided by integration into the customs union, that any community integrates and positions itself both separately and together. By the fact that there is a good experience of the European Union, we are paying attention to the experience of and knowledge gained in the US, in Japan, in BRICS companies that are compiling right now. So, undoubtedly, each company. And once again, I would like to underscore the idea that was mentioned yesterday. We are in a unique place. We have the potential to take the best, and against this background, to offer ideas to move forward the development of the pharmaceutical market further. Many professors took part in these discussions. This is how the scientific knowledge is developed. Any new scientific move is not happening in a vacuum. It's based on previous findings. We're just analyzing the previous experience, taking the errors and moving ahead. Recently, I had a talk with the Deputy Minister of Development and Industry, and he told me the following: if we don't push pedals, bicycles would fall. So I want to push the pedals, to pedal up, to move ahead. And I would like to say that I appreciate your participation in the conference. As the health protection will be always taking the leading edge, this area deals with everybody, with all of us sitting in this conference hall. Because nothing in this world is more precious than the human life, so short and so valuable, because everyone who lives on this planet leaves a mark in history, and everyone wants their mark to last longer. And I think that any conference of this scale will bring us closer to the solution that the health of citizens is in good hands. Once again, I am extending a thank you to all participants, and I hope that the work will go on, and that we will continue working tomorrow.
Спасибо. Спасибо, Елена Анатольевна.